Okay. So the instructions here walk you through where you can find the restriction analysis challenge handout in the PowerPoint and that kind of thing. That was, again, part of topic 3's module. So we will go through the challenge, and then you are going to, or I will pick up for chapter 15 for translation. I have the syllabus pulled up, and pretty much what we're going to end up doing is I'm thinking just bumping everything by 1 week, because the way that it works out, we end up having some classes that don't have lecture chapters associated with them because we're usually working on our projects at that point. So I think we'll be okay if we end up pretty much just skipping the week and keeping on schedule otherwise. The only thing that I worry about is exam 1 for topics 1 through 5. I don't want it to get pushed to after spring break because we've already so far and you haven't had a test yet. So that will probably go ahead and be done during the same week that it's already set on, because it's topics 1 through 5, and you'll notice that this is topic 6. So then we can just kind of finish out right before spring break through topic 5, and then we can pick up topic 6 after spring break when we're back in the classroom. It's just kind of what it's going to have to be. So this time I'm going to be going over the restriction challenge that we didn't get to talk about last week. On Wednesday, we will go over transgenic animals, box P2. I'm going to give you a video about RNA folding in the same way that I did the videos for like the recombinant paper plasmid DNA scissor stuff, because it's, again, one that you'll print out and you can fold the little strips of paper and make the 3-dimensional tRNA molecules and stuff like that we'll talk about tonight. Because we aren't going to be at school. I mean, there's some virtual labs about digestion or about gels, but you can't really do the gels. But you'll have the restriction challenge to work on still too. The following week is for PTC testing. It's where you're going to actually extract your own DNA from your cheek cells. So we can't do that until you're back in the classroom to be able to do the DNA stuff. So what we might end up doing, so model organisms is something where you just kind of look at a lot of different kinds of critters. And we can at least look at videos of all of them, and then we can play with them once you get back into the classroom. But I think that way we aren't losing lab time completely, because if we are going to plan to do projects, we should start discussing them now over these few weeks, and then those will be like, if we need to order anything, I'd like to have it ordered before spring break so we have time for shipping and stuff. I was not expecting to get pushed online, so it'll be an exciting adventure. So, all right, so the restriction challenge stuff, the PowerPoint is already pulled up down here, and the handout for it, I'll just show you here. You could download it if you have not already. Has all of the maps. These little maps are going to be shown in the PowerPoint as we go through. We'll have P target, P source 1, P source 2, and then a linear one to look at. So, all right. Actually, do here. Okay. So, restriction challenge. This is tying together everything you learned from DNA scissors and recombinant paper plasmids and from P-Glow. So our challenge on the first one is to move the gene from miraculin into an expression vector. So miraculin is a protein that is produced by a berry that if you eat it, it makes things that are normally really acidic taste sweet. So you could actually get it in little pill forms and they last for about an hour. It's just an interesting thing. It has nothing to do with this apart from the fact that it's a gene that you can move around. And an expression vector, a vector is a plasmid, so a circular piece of DNA that when we say something is expressed, we mean that the gene is turned on to produce the protein. So an expression vector is just a way to carry a gene that you're going to want to turn on. So here is our plasmid map. The first one has the gene for miraculin here in red. We have a promoter in purple and an origin of replication in gray. So question 1, what enzymes would you use to cut out the gene for miraculin? So we can see here that we have PST1 and then you have other genes down here, or not genes, you have other cut sites down here. We have to pay attention to the fact that if you used EcoR1, there's another EcoR1 cut site at the 1000 base pairs. So you would end up with extra cuts that you didn't want. You also don't need to include the promoter on this one. And when we go over to the target one, we're going to show that there is a promoter already in the expression vector. So we are going to want to use PST1 and not 1. Because again, EcoR1 has 2 cut sites at 1000 and 1350. So we don't want to cut in the middle of our gene. And we don't want to use BAMH1 because that's giving us a bunch of extra stuff we didn't need here. And again, this one cuts in both places. So now, what size fragments would you create if you cut P source 1 and P target with PST1 and not 1? So to look at this, we have on your PDF, if we cut with PST1 and not 1 on both of these. So the first one that we've already talked about would be an easy one, that the difference between 400 and 1200 is 800. That's going to be our gene of interest. And the backbone piece would be the 3000 minus 800. So it'll be 2200. If we go look at the target, we haven't talked about it yet. It has an origin, a gene for ampicillin resistance. We have a promoter. MCS stands for multi-cloning site. It just means it's a site that has a lot of cut sites in there. And then there's a LAC-Z gene that we're going to talk about later on, why that one's important. But if we're doing PST1 and not 1, 950 and 990 is going to give us a difference of 40. And then you do the whole big piece minus 40. So you should get something like this, that you would have a piece that was 800 and 2200 left over. And then on the target, you would have a 40 base pair piece and a 5460 base pair piece. So unless y'all speak up to tell me to slow down or to ask a question, either in chat or to unmute yourself and ask, I'm going to assume that you're okay with this. We're also going to show, like in this case, PST1 and not 1 are sticky ends. So that's why it shows this little overhanging region. That's going to be important later on when we start putting them back together. It cannot go in the wrong way. Not 1 has to bind with not 1. PST1 has to bind to PST1. It can't, you can't reseal itself back without having it anything like that. Assuming that only plasmids with a single origin of replication are possible, which of the following products will not be made when ligase is added to the mixture? So we're assuming that we are putting all of these in one container. One test tube is going to hold all the different cut up pieces. And we're going to say when we add ligase, that is our enzyme that helps glue things back together, which ones are not going to be made. So we can see we could make A because we could put our 2200 back with the 800. We could have flip-flop the 800 and the 40, or we can just religate the other one back to itself. So 6300 is the odd one out. We wouldn't get that one. If the growth medium contains ampicillin, which of the following ligation products could you detect inside the colonies of transformed bacteria? More than one answer may be correct. So the important thing here is that the ampicillin resistance gene is on that 5460 base pair piece. So we looking at the numbers we had here, from the answer choices we were given before, the ones that are the 2000s, those don't have ampicillin resistance. And then that gives us two possibilities. The one where the 40 base pair just went right back in, and then one that has an extra 800 base pair because that's now carrying the miraculin gene. But that means that we'd have two colonies, and we want to be able to tell them apart on the plate. Miraculin doesn't have a color associated with it, so you can't look at it that way. It's not going to glow in the dark or anything like that based off this vector. So we want to find a way to distinguish between the two potential isolates, the regenerated p-target vector, that's where the 40 base pairs just went right back in, and the recombinant miraculin containing vector. It says to use your gel boards to show the products from your restriction analysis, but this is assuming you were in the classroom and that I could see what you were working on. We'd have these gel boards that are just kind of a template for you to be able to draw the bands on. So we're going to go back and look and see how you could tell which ones were just p-target with itself or the p-target with that 800 base pair. Let's go look at the pictures here. And we can see that there's some other cut sites to look at. I'm going to show target. So to guide you a bit, what restriction enzyme would you use to create unique fragments that would help distinguish between p-target and the recombinant plasmid? So if we look at these, let me go ahead and show the answers here. P-target being that the 40 base pair just went right back into it, and the recombinant saying that the 800 base pair went into it. So while all of the options would work, it might be hard to tell a 5,500 base pair one apart from a 6,250 base pair on a gel. So it'd be better to pick one that's going to give us some more interesting options. So if we did any of these that are a single cut site, so that's A, D, E, F, and G, they would all show the same banding patterns in terms of there'd be 5,500 or 6,250. Because they cut one time, it's just turning that rubber band into a straight line. If you cut with EcoR1, we would get some variations here. You'd only have one band for the P target, but you would now be able to get two different bands for the recombinant plasmid. If you do CIA1, you would actually get two bands versus three bands. And so that's one to pay attention to because it had cut sites in more than one place. I'm sorry about that typo. Need to have space in there. So I mentioned before that LAC-Z operon, or the LAC-Z gene that comes from the lac operon. And you can use this as a kind of cheating step. I mentioned that miraculin doesn't have any color, But lac-Z does if you mix it with a chemical called X-gal, which is the type of sugar that when cleaved turns blue. So if you have your gene for ampicillin resistance and the gene for beta-galactosidase production, that is lac-Z, and you're trying to insert foreign DNA with sticky ends into the middle of that blue gene, and it's going to break it. So if it works, you put it in and it has the foreign DNA inserted, you get cells that will grow on the plate because they are resistant to ampicillin, but they don't produce any color because that blue gene is broken. It won't be able to cleave the sugar because lac-Z is broken. Now, if you put in the one where like the 40 base pair just religated back in and no foreign DNA got stuck in, you get the second set of plates, which is where the bacteria are going to grow because they're ampicillin resistant, and they're going to be able to produce a blue color because the lac-Z gene is intact. It wasn't interrupted. If you had cells that didn't pick up either the plasmid, they can't grow because those cells are not resistant to ampicillin. They're going to die. So that usually if you did this in real life, you would get a plate like the one shown on the right here that shows the blue colonies. Well, those are ones that lac-Z is working perfectly fine. That means it did not get our insert. We don't want those. We're going to completely ignore those blue ones. And we would just pick up the white ones because that shows that there's foreign DNA inside that gene. So that is kind of a cheating step of why you would actually use this expression vector. But we're going to show you a problem with it for part 2. Do I have any questions over part 1? So all we're saying is, could you cut it up and reglue them back together? Now, challenge 2 uses P source 2 as its source, and we're still putting it into P target. So to cut the gene, what enzymes would you use to cut out the gene from miraculin? So we can look, and now we have 2 choices. Eco R5, Hey3 are right here at the beginning, and then small one is down here. So I'm going to tell you the secret now is to go look at P target, and because you could go, let's see, what was one? Small one. If you went Hey3 to small one, the insert would, or the piece would be the right direction. But if you put it in with small one and Eco R5, it would now be upside down. And your gene is in reverse, it can't be read from that promoter. So this week when we're talking about transcription and translation, we know that the order of codons is going to be important. And so having a piece of DNA going the wrong direction means it's never going to get read properly. So we want to make sure that it's going to get put in going the right way. Now, what size fragments would you create if you cut P source 2 and P target with Hey3 and small 1? And so those are the answers that we just decided that we wanted. And you can always go through and do this. I don't want to run out of time, so I'm going to keep moving forward and show the same thing, that we would have a piece of 800 base pairs that has our miraculin gene in the backbone that is just extra crap. And then on the target, we would have lost out 50 base pairs. And then we would have our backbone that has our ampicillin resistance gene and everything that was in it for the first part. Now, there is something different on these. You notice the image before, they were like edged, like there was a little notch on them because there were sticky ends. These are blunt cutters. So the problem with blunt cutting is that you can put them back together and it doesn't matter what they're going to. So the miraculin gene could go in the wrong direction now. That's going to cause some problems. So assuming that only plasmids with a single origin of replication are possible, which of the following products will not be made when ligase is added to the mixture? And we can see here our options just like before, but the E wouldn't be made because it would have two origins. And if, again, just like before, if you use growth media containing ampicillin, which ones would you be able to detect? More than one answer could be possible. And so we can rule out A and C because they're using the 2200 base pair backbone that doesn't have ampicillin resistance. But you now have three options for possibilities. One of them is that you inserted the 800 base pair miraculin gene. That's B. Then there's D, that is the 50 base pair got put back in. And then E is saying that the 50 base pair didn't go back in and it just resealed itself completely. That is our troublemaker. Well, technically B and E are troublemakers because when we had our sticky ends, we could not, or we didn't have to worry that the insert was going to be put in the wrong way or something like that. Like it could only go in one direction because knot one could only bind to knot one and PST1 could only bind to PST1. Blunt end cutters, however, are shown like Eco R5 here. These guys don't have an overhanging region they need to line up with. They can just get stuck together. So you see how here, if that piece didn't get inserted, you could just stick this right back to itself. And now your blue-white screening isn't going to work because that blue-white screening is running off the idea that you had a functional LAC-Z or a non-functional LAC-Z gene. If you insert the 50 base pairs back in, you have a functional LAC-Z gene. But if you just glued it back shut without putting an insert at all, you would now have a broken LAC-Z gene, but it wouldn't, so it'd be a white colony, but it wouldn't be the one you wanted. So there's some other ones that are possibilities because you, like before, we could only get very specific answers because of that, the overhanging regions. Now, if we stuck these together, you could get lots of crazy things. You could get that 50 base pair thing going in multiple times and just some weirdness. It wouldn't necessarily come out to just two parts sticking together. So our other issue is because that miraculin gene could go in either way. We now want to find a way using gels and restriction enzymes to tell which direction your miraculin gene went in. Now, I have a list here of showing the regenerated p-target vector. That is where the 50 base pairs is inserted. The self-ligated p-target backbone, that's where there's no insert, it's just in by itself. And the two recombinant miraculin containing vectors, that's showing that you put in the miraculin gene one way and then you put it in going backwards. And how would you be able to detect them? You don't want to use the exact same enzymes because if you cut out the miraculin gene just as it is, you wouldn't have been able to tell which way it went in. You'd have a band on the gel and it was like a band that says 800 base pairs. You're like, yay, it had miraculin in it, but which way? So find a way to cut it that you're going to see a difference on the gel based off whatever enzymes you're using. There's, again, more than one correct answer, but you need to go look at all the options that are available, specifically the ones that are going to cut uneven amounts to be able to go to see them on the gel. So that is your homework for this one. Now, it is a group-based thing. So I just want to make sure that everybody understands. If you go to your People tab, and mine's going to look a little different, but I have the extra teams set up, like Monday small teams. And I'm just clicking on one of them, like Monday 1B, and going to the group, visit the homepage. This is a workplace for you. You can upload any files you want. You can create pages, which is just like you can type your title, you can type in anything you want here. It's kind of like Google Docs, except it's not real time. It's after somebody has made changes, they click save, and then it's accessible to everybody else. Collaborations, however, is pretty much using Google Docs within Canvas. So you can use that if you want, but that's going to require everybody to have a Google account. And I don't care if you use regular Google Docs, whatever, just as long as y'all have a way to communicate with each other, because you want to just come up with one answer for everybody. You don't want to each do all the work again. So know that this is here under the People's tab. What was it for that part? Oh, then there is part or challenge 3, where you have been asked to map a linear piece of DNA. You set up 3 different reactions in which you cut the DNA with enzyme A, enzyme B, and then both A and B. Both enzymes cut twice. Draw a map of the DNA with the locations of the cut sites indicated. And here are the results from the gel. So this is 1.5 thousand base pairs. So 1.5, 2.5, 5.6, 3 and 3.3. And then if you cut with both enzymes, all of these come out. So you need to figure out where the cut sites were for each of the enzymes. I'm going to show you my board. So for example, if you had, let's see what the size is, 1.5, 2.5, 5.6. So 1.5, 2.5, 5.6. They are 1.5 piece, 2.5 piece, and a 5.6. Now you don't know, was it one line of that actually went in order, or was it that they went in a different order? Like here we could have it to where there is a cut site here and a cut site here, where this was at 1.5. Its address. And this one was now 1.5 plus the 2.5 together, so it would be 4.0. Like, did it look like this? Or did it happen to have that maybe the 2.5 came first and then that 5.6 went in? If that were the case, now we'd have 2.5 and 2.5 plus 5.6 is 8.1. No, we don't know which ones it is. So you have to play around with these, and I recommend literally taking pieces of paper that are about the right size, like if you have graph paper or lined notebook paper and you cut it sideways, however you want to do it, because you need to figure out, you can't do it just based off one cut. You need to now look at your B's and when it was cut with both enzymes to find out where all the differences were. Just like we did with P-Glow, you were able to do the math for that. Now, it's going to throw you off because enzyme B here, remember it says it cuts twice? If you took a linear piece of DNA and you cut it twice, you should have 3 bands. That means that one of these numbers is a repeat. So you have to figure out which one it is. Because if you had, like on this picture here, another 1.5 piece, it would land on top of each other in the gel and it would just look like one band. So that's what's happening on one of them from the enzyme B. There are a double amount of one of them. You can help figure it out by add these up to find out what their total is, add these up to find out what their total is, and that'll tell you what you need to do to figure out which one's missing here, who is actually a double agent. Okay. Does anybody have questions over this? If not, I'm going to pause this recording.